My name is Erica Holzbaur, and I have a confession to make. I'm a cell biologist, and as a cell biologist, it's literally impossible for me to talk about mammalian cell biology in 1 hour. But I crammed as much as I could together, and we'll see how it goes. I tried to assume that you haven't had a lot of biology, some of it in the room, and others have had more. But hopefully people have had high school biology. Yes, ring a bell? Okay, so we'll start from there and build on it. What I want to do today is not give you specific facts or figures, but get you to understand some major concepts of our current understanding of cell biology. So as you'll see right at the top, if you learn nothing else today but that compartmentalization and dynamics are two key principles of cell biology, and if you see beautiful pictures of cells in movies, then we've succeeded. But we can only really succeed if you ask me a lot of questions and interrupt if you don't understand something. So I challenge everyone to come up with a question. Erin will lead off. So I divided the talk today into the first theme where we'll talk about compartmentalization within a mammalian cell. We'll move on to dynamics, and then finally think about specialization, because there is no such thing as a generic cell or even a generic mammalian cell, and we'll think about how different they could be. So just to place us on the scale, you've been having a lot of talks in different types of order. Some have been focusing more on the molecular end of things, and some have been more probably towards the tissue end. We're going to be here right in the middle, sort of centered on organelles, which is sort of the focal point for cell biology. It's within the context of different types of cells. We're looking at organelles and how they function as basically micro machines to keep the cell working. We're able to do that with a really molecular understanding because obviously advances in DNA technology and cloning and ability to knock out genes and put them back in. So we do have a molecular understanding, but our major focus is sort of in this molecular understanding of cellular organelles and then how they function in the cell. Where the field is going and where it'll be when you are out of school, we'll be then thinking how these cells really function in context with the same molecular understanding. Oh, thanks. So to start with compartmentalization, we'll go back to high school biology where, and I know you did this, I have 2 kids and they went through school, you made these pictures in grade school where here's the cell and here are the organelles. So you've seen this kind of picture before where you had the limiting plasma membrane, you had the nucleus, which is obviously the largest organelle, and so anyone can identify that. Stretching out from the nucleus, you have the endoplasmic reticulum, the Golgi, mitochondria, which we'll talk a lot about. So I'm not going to rehash all that and go through all these organelles, because what I really want to emphasize is 2 major features. One is compartmentalization. Why are there these different organelles? If you think about it, it gives this cell an ability to concentrate specific biosynthetic or degradative processes in specific compartments to keep them walled off. Part of that is safety. For example, the lysosome is a degradative organelle that breaks down proteins and other molecules. It requires pretty powerful enzymes that are clipping away at proteins. You obviously wouldn't want those enzymes running amok in the cell, so by concentrating in the lysosome, you're able to do the job, but keep it sequestered from the rest of the cell. Similarly, the nucleus is where you store the inheritable DNA. You want to keep that safe from the rough and tumble world of the cytoplasm, so by sequestering it in a membrane-bound compartment, you're able to both keep it safe and also concentrate the enzymes that are going to pass on the genetic information or transcribe it for RNA that'll be then sent out to the cytoplasm. So each of these compartments allows the cell to function in a very high efficiency type of way. But what it means is that none of this is static. As you learn this picture, that's not really a cell. What's happening in the cell is you're getting flow between these compartments all the time. So a perfect example is when we think about the endoplasmic reticulum. In your classic picture that you learned in high school, it's this membrane that's contiguous with the nuclear envelope that bounds the nucleus and then extends out in this complex array where you find ribosomes, which are going to translate proteins. So if you look, that's the schematic, and if you look at what the ER actually looks like with a dye here, you can see that it actually extends throughout the cell. And if I had a movie of this, you'd see it's incredibly dynamic over time, continuously changing and remodeling. So it's a distinct organelle, but it's also continuously in flux. So I also wanted today to emphasize the new developments in our understanding. Cell biology is an incredibly exciting time right now because new technologies are being developed that allow us to look at things in more detail and more specificity than we ever could before. So a lot of that is live cell imaging, which we do a lot of in my lab. Others of that is super resolution microscopy. And this is a paper that just came out last year where one of the major leaders in the field used new advances in three-dimensional super-resolution microscopy to look at that same ER structure we were just looking at in a flat image. And here it's reconstructed in the 3D volume, and you can see these beautiful, gorgeous 3D structures of the endoplasmic reticulum. So why is that important? You can say, okay, it just looks like a lot of blobs to me. Well, classically, when people talked about the ER for decades, and Rick will agree with me here, there was this dogma that there were two different kinds of ER. There were tubes and sheets, and that these were very different. Well, it turns out they were only different because of a resolution problem. And as soon as you could look in more detail, you saw it was all this beautiful tubular array. So basically, by higher resolution analysis, we could then understand the structure and the function, because we know that there are proteins that give it this particular structure and allow it to remodel over time, and that with a better understanding of the structure through modern techniques, we can understand the function of the organelle. So I told you there's flow in cells, but I'm still showing you static pictures. What happens as these organelles interact? Well, we have this ER that I was showing you, and these diagrams looks like it's just localized, but as I showed you, in reality, it's throughout the cell. But you'll get trafficking of vesicles, specifically from the ER to the Golgi. So what does that look like in reality if you look in live cell? It looks like this, where here we're looking at something that's loaded in the ER and is going to bud from the ER. And as I told you, the ER is not just one specific localized membrane. It's actually all throughout the cell. But the Golgi is more discrete in many cells and a more unified, closer to the cell center. So in this cell, here's the nucleus, and there's the Golgi. So once I show you this movie, what you'll see is the actual process of vesicles moving from the ER to the Golgi and accumulating there. That's what you can watch, and you can watch this beautiful movement driven by molecular motors along the cytoskeleton. What you're not seeing so well in these movies, but is equally exciting, is this beautiful budding from one compartment to form these organelles that then move to the Golgi and then fuse with the Golgi and bud there. So what we're seeing in even just this brief movie is this idea that you have separate organelles, but you have continuous flow from one to another. Does that make sense to everybody? Yep, great.
How do they move? Are they like shot or?
Oh, I will show you exactly how they move in a little bit, if you could just hold that thought. But briefly, as I'll show you, and I think Ram Dixit already showed you, there's tracks all through the cytoskeleton. Microtubule tracks and actin tracks, and Brianna's laughing because it's exactly what we study. But there's microtubule tracks, and these vesicles have bound to them molecular motors that walk along those tracks. And we'll talk about that in a few minutes. Great question, so keep it up. And I just put this in here to show that even something that looks as non-specific to the eyes of maybe an engineer, when you actually look at the Golgi, for example, you can see that each part of it is different. It's polarized with budding at one face and It's basically a factory where you get modification of proteins throughout in a very vectoral process, and then sorting at specific stations. So really, it's all a finer way of engineering, even though it looks like, just lipid-based organelles. So I think that's what's really exciting about cell biology, is trying to understand how these machines work so well. And then again, what I was saying, you get processing through these stacks, and then you can get sorting where different codes are read to take things to different outputs. So you can get the specific generation of transmembrane proteins, which will be the adhesion molecules that Rick Kassoyan is going to tell you about. You could get the packaging of things that are going to be released from the cells, such as neurotransmitters or secretory hormones in these vesicles that will then release their contents, or you could get trafficking to degradative organelles. So again, flow from one compartment to another is continuous, and it's a smart flow. It's not just a single pathway that just goes out in one direction. You get this specific sorting to cellular destinations. So here's a further example of that, where you can see these release of these, I forgot exactly what this is, I don't know if this is a neuro slide, but neurotransmitters, for instance, or a hormone released here. You can see, do you remember, Rick? I don't know. But specific release in an exocytosis process. And not just the specific proteins to be released are highly regulated, but where they're released on different faces of the cell is something to think about. So all of this is highly organized, and if we had time, which we don't, we could talk about the specific signals that take it from one face of the cell to the other. But as you're thinking about a cell, it's not just a bag of stuff. If it's in context, especially in a tissue, it can have very different faces, one versus the other, and so it really matters if you're delivering things to one face or another. Yeah? Yes, it can, actually. So the long-range movement will be along microtubules, and then some of the short-range movement is via actin filaments, and then driven by myosins. So you can get handoff of those things. Yes?
Is this new? Because in so many textbook images, you never see any of that. Like, even in the picture here, it's just kind of a vesicle movement.
Yeah, so people are very interested in the localized role of actin and myosin and moving these organelles into position. And there is a lot of ongoing work there. The general concept is not new. It's, I'd say, 10 or 20 years old. The idea, and we'll talk about long-range movement in a minute. If you want long-range movement, that's along a microtubule. And then if you want short-range tethering or localized movements like ectoplasmic membrane, then that would be actin-based movement by myosin motors. So just to sort of continue with this cycle idea, you take things, ship things out, but you also have to ship things back in by endocytosis. That's one of the major ways that cells stay in contact with their environment. Uptake of material from the plasma membrane to form endosomes that are then cycled through these same membrane pathways. And it's not important that you remember any of these specific pathways right now, but just get the idea that there is continuous flow through these pathways and that it's non-random. It's highly directed, not to a specific spot, like it. This doesn't have to go to a specific late endosome or a specific lysosome, but there is molecular identities to each of these different compartments that allow these the targeting of these organelles. So just the process of endocytosis, I threw this in because this process is beautifully understood, and also again shows you that juncture where we have molecular detail that shows the formation of a molecular machine that can drive this process of endocytosis. Here's the plasma membrane, and you can see clearly from this electron micrograph the formation of this coat then causes more of an invagination until you get this tight neck here that's pinched off specifically in an enzyme-driven process to give you your endocytosed vesicle. And we can define all the different components that allow this molecular machine to occur to uptake important molecules. So what type of molecules are taken up? Well, you've all heard of cholesterol and LDL and such. That process uses this type of endocytosis, as does neurotransmitters and recycling of neurotransmitters, so for your neurons to function. Trophic factors, many, many different things. It's a major way for cells to connect with their environment. And finally, the end process, as much as cells make new things, they have to break things down, and that's in this degradative pathway, which was one of the things my lab's gotten very interested in, where things either coming in by endocytosis, or if you have an aging organelle, anything that has to be broken down, the cell then breaks it down and recycles the components back for reuse. So that is a huge overview of flow through the cells. I want you to get some concepts out of it, which one is this continuous movement from one compartment to another. You never see a static cell. Always you have this flow going on from compartment to compartment. Another concept is in these flows, the membrane topology is always maintained. If you have a protein with a cytosolic face, that face will be cytosolic whether it's on a vesicle or on the plasma membrane. So in all these different budding and fusion events, topology is maintained. And that's really important because sometimes you need to maintain an inside that's distinct, or most of the time you need to maintain an inside that's distinct from an outside. And the perfect example of that is this lysosome that we were just talking about. This is the organelle that breaks things down, and it's got an array of things meant to degrade nucleic acid or proteins or lipids. The cell's got a great mechanism to regulate these. These are all called acid hydrolases because they're most effective at an acidic pH. So if you keep the lysosome at an acidic pH, all these enzymes will function. If they happen to leak out of this lysosome, which can happen, especially in aging cells, they'll be in the cytosol, but they'll be less effective because you're not at the correct pH for these enzymes to function. So topology is important. Specificity of targeting is important. I'm telling you that from one organelle to another, you have this continuous flow, and it's via the pinching off, the budding of transport vesicles that will then move, and we'll talk about that, how they move in a couple minutes, and then fuse with another organelle. But there's a specificity to this process. These will not just fuse willy-nilly with any compartment. There's specific molecular identities, proteins called snares, that mediate this specific docking. So it's very important that you get the flow, but it's very important that stuff go to the right place. So there's molecular detail known in the pairing of these vesicles with their target membranes. And so again, coming back to this flow, you've learned in high school biology there's an ER and a Golgi and this and that, but the important thing is they aren't so discrete in the sense that they never change. There's this continuous movement of material both forward and also back. If everything just moved forward, you'd wind up depleting this compartment of lipids and such, and you'd wind up with no ER and everything over here. So you get this flow of vesicles back to restock your compartments with lipids and protein components. The other exciting topic that's emerging and is distinct from what you learn in biology is not just that these organelles can fuse with each other, but that they can interact physically with each other and affect each other's specific properties. So one really exciting area is coming again from this super resolution microscopy, where we can look at the ER, and this is a super resolution micrograph, and the ER is in green here, and in purple what you see are mitochondria. So when you first look at it, it just looks kind of a mess. But when you zoom in, what you can see is there's specific contacts of ER with mitochondria. And here the colors are a little bit changed. But you can see in this case it looks like the ER is wrapping around mitochondria. And what we happen to know now is that where that ER wraps, it's actually enhancing the fission of the mitochondria. So it's that contact is reshaping a different organelle. And there's a question.
Yeah. So are they using specific membrane markers?
Yes.
Is it like a protein marker, or is it like a stain?
Yeah, so that's a great question. So classically, cell biology was done in a static way, which is why we had a static idea of it. You'd fix cells, meaning you'd glue the proteins basically in place by cross-linking them, and then you'd put an antibody on and then look with a secondary antibody. And so you'd have beautiful pictures of what is in the cell at any given time, but not this idea of flow over time. What's been a revolution in cell biology over the last, I guess, 20 years by now, is the idea that we can make fluorescent proteins. Either it started with fluorescent dyes that would go to maybe a lipid compartment, and now it's fluorescent proteins that'll specifically target different organelles. And so we can watch them either in fixed images at high resolution, or, which is what this is, a super resolution, which this was a fixed process, or this was from a live cell movie, where you can watch fluorescent dyes that are specifically targeted to mitochondria, or here we're looking at actin and ER in the red. It's a little bit hard to see here. So we have a very dynamic view of it now. Great questions. Keep them up. And just to show you how this is exploding now, a few years ago, 4 or 5 years ago, even the best labs would do two really nice, two separate colors in live cell microscopy. So you could look at two things. So for instance, here, you could look at mitochondria and you could look at actin, but you couldn't also look at ER because there were just not enough good specific fluorescent probes and not enough ways to separate the channels so that you could clearly see one signal without having it spectrally overlap with another. But in an astounding paper that just came out this year, one of the leaders in the field, Jennifer Lippincott-Schwartz, has now got an approach where you can do live cell microscopy with 6 different colors simultaneously. So you can simultaneously label lysosomes and mitochondria and ER and peroxisomes and Golgi and lipid vesicles. And you can see them all at the same time and then really ask these questions about how they interact, how they overlap. So in this case, when they did an interaction map, they showed the highest interaction was between mitochondria and ER, and that's for what I just showed you, that we now know that ER is there to help pinch the mitochondria, to fizz the mitochondria into separate pieces. So this is exploding, and this is exactly in the area that you all are interested in. This is a very computationally based approach to improve microscopy by separating out the channels and cleaning them up after you take the images. So this area where you're thinking about engineering and math and biology is super exciting right now. This is just more images from the paper. I just really liked it, where you can see in 3 dimensions these multi-labeled organelles. All right, so that in a whirlwind gives you sort of an overview of trafficking through the cell. And this is the major advance that makes a eukaryotic cell different than a prokaryotic cell. So bacteria wouldn't have all these internal membranes and wouldn't have this ability to compartmentalize and have this complex biosynthetic and degradative pathways. And the leap to eukaryotic cells, both the plants that Ram told you about and the animal cells that I'm talking about, have this ability to compartmentalize all these different functions. So now we're going to move on to theme 2 and dynamics, and now we'll get to what many of you were asking about the actual movements from one place to the other and how that works. I understand that several people have already introduced you to the cellular cytoskeleton. I'll reintroduce you because I'm obsessed with it. But basically, in animal cells, you have three major flavors of the cytoskeleton: microtubules, actin filaments, and intermediate filaments. So as soon as you hear the word cytoskeleton, it has sort of an unfortunate connotation because you start to think that these are very rigid. And again, you look at these pictures and they look so rigid and fixed in place. So you're thinking, oh, it's not very dynamic over time. But what I hope to convince you is, at least when we talk about microtubules and actin, we're talking about structures that are continuously dynamic and allow the cell to both carry on its regular functions and also respond to its environment. Intermediate filaments are a little bit different. They're more structural elements, so they're often much less dynamic than the other two forms of the cytoskeleton. So they are important, but they're also usually overlooked by people that are excited by dynamics, and I'm going to do that also and focus on microtubules and actin. So first, actin, which I think Ron talked to you a little bit about, and Mike Ostap. It's formed from a monomeric protein that then assembles into a filament like this. Because the protein is not the same at either end, and because it assembles in a head-to-tail way to form a polymer, that polymer is polarized with two different ends. And the most important thing you can learn about the actin and microtubule cytoskeletons is that easy fact, that they assemble into polymers and the two ends are different and have very different properties in terms of assembly dynamics and disassembly dynamics. So this is a very dynamic and highly regulated process in the cell. Any given actin filament could be a structural one that could last for quite a while. We'll talk about where you'd find structural elements, or it could be very dynamic and a very transient type of polymer. But we know in detail the process for actin assembly. It can happen spontaneously, but it rarely does in the cell. It's almost always the assembly is driven by nucleation promoting factors that give you regulated assembly, and the disassembly is often tightly regulated. You have enzymes that come in and clip the filaments and enhance their ability to fall apart quickly. So in a cell site, and here I'm looking at a neuron because it's large and beautiful and has gorgeous actin staining. Where would you find actin? Well, if you look at this neuron, this is what's called the cell body, basically the very center of the cell where you have the nucleus and much of the ER and Golgi elements. You'll see there is actin, but mostly associated with the outside membrane of the cell, the plasma membrane. Where you see the actin highly enriched are at the tips of these processes. The dendrites here, or the axon, which is this long extension, at the very ends here. And that's not by accident, that's for a specific purpose, because actin, as you can see in this movie, is a live cell image of actin in the end of a growth cone. You can see all these dynamics of actin driving forward this extension of this process. So when your nervous system is developing, the actin at the growth cone is leading the way and pushing out the membrane. And you require those continuous dynamics of actin polymerization and depolymerization to move forward over time. So that makes actin sound very exciting and dynamic. But we also know actin has a major structural role. And again, this is a new development from super resolution microscopy, which I sound like an advertisement. I'm sorry about that. But up till a few years ago, if we were looking at neurons, and we want to look at actin in green here, you'd have this kind of image, which is a standard epifluorescence image. It's interesting, but hard to get enough detail out of there to really see something. But when you go to super resolution microscopy, and in this case it was three-dimensional super resolution microscopy, then you can start to see some really interesting things. So you're not convinced. You're saying it still looks like spaghetti to me. But if you look at this image here, and the actin staining there. Is that still looking like spaghetti to you, or you're starting to see structure? So just a few years ago, somebody was looking at this, and I thought they'd see just actin at the tips, and then they started to look along here. I was like, wow, I see something. I see a regular structure, right? If you did a line down there, right, you'd see regular intensity increases, regular structure along there. That was brand new. Nobody had a clue that that structure was there until they looked at these pictures. So here we're zoomed in, and you can see these actin structures all along here, and these diagonal lines, which are actually, in these lines, which are actually rings. And then they thought, I wonder if that's giving the whole axon, which is a long, extended process, I wonder if that's giving it structure. So they looked at a protein called spectrin, which is known to interact with actin in red blood cell membranes. And they said, well, I know actin can bind to spectrin. Is spectrin also highly organized here? And look, it is. And it's intercalated, one versus the other. And then adenosine is a protein that's also involved in this interaction, and that also shows the same pattern. And maybe it's a little like imperfect because it's biology, but you can clearly see this repeating structure, and you can quantitate the differences and come up with an entire model based on these beautiful pictures in which you have this structure of the actin rings, and then spectrin spanning them to give you this regular array. So brand new, exciting biology that's critical to understanding how neurons with these long projections, which as we'll see can stretch a meter, are stabilized by actin and interacting proteins. So from nothing, a clear model that's very much like, I don't know if you guys ever vacuum, but like the vacuum tubes, it's the same kind of structural model is used by your neurons.
Does that have anything to do with like the charge carried? Because you said actin has like a positive.
Oh, in terms of signaling, no. This, as far as we know, has to do as a structural element to stabilize this structure. It doesn't have to do with neuronal signaling, but it was a good question. So actin is one form of cytoskeleton. Another major form that we talked about was microtubules, and their subunit is tubulin, but not just a single molecule of tubulin, two of them, an alpha and a beta tubulin that come together and make this structure like this. So this is one polypeptide, and this is another polypeptide. They're not covalently bound, meaning you can separate them out. They're non-covalently bound, but in a very tight association. In the cell, you would never see one without the other. They're always tightly bound. And together, this forms the monomeric unit of a polymer once they come together. But again, the same thing is true as with the actin subunit, because this end of the monomeric unit is different than this end. That means once they assemble into a polymer, it's polarized. It happens, though, that tubulin assembles into a much more interesting filament than a microtubule. I'm biased, but it is, because it's an actual tubule. So you could see head to tail these tubulin dimers line up, but it's not just a single strand. It's actually multiple strands, somewhere around 13 protofilaments. It can vary a little bit depending on the species of the microtubule. But you get multiple protofilaments assembling to form this hollow tube. Again, the whole tube is polar. One end is very different than the other because the two ends of the tubulin dimer are different from each other. So the properties of the beta tubulin end are very different than the alpha tubulin. So this microtubule had very different properties. So I shouldn't even show this. I should get you guys to guess. So you're engineers, some of you, or biophysicists. I told you actin forms a single filament and microtubules form a tube. So what kind of properties are going to be different between the two of them? Which is stiffer? Yep, just exactly the reason you'd think. Actin filament would be rope-like, and this is more like a cardboard tube. stiff until you actually bend it, and then it falls apart. So these things have been measured. We know actin filament's thin and flexible. Microtubule is much thicker and stiffer. Those properties can be changed in the cell. I told you that actin was used in a structural way in axons. The reason it can be used in a structural way is you don't have a single actin filament in those cases. You start to get bundled actin filaments. So you build up an array of actin, excuse me, and then it can be a thicker structure that can be stiffer. The most important things about these two, I keep telling you, are polar polymers with two different ends that have different properties of the different ends, kinetics of polymerization, depolymerization. The other thing to remember is that these are both really active polymers, that they can continuously grow and shrink in cells. So just to show you that, here we're looking at a movie of an in vitro experiment in which you take a microtubule organizing center. We don't have time to go into that. But you put it in a solution that's rich in tubulin. And some of us were just talking about how you get tubulin from cow brains and pig brains that you get at the slaughterhouse. But basically, it's relatively easy biochemically to isolate tubulin. You put this organizing center in there, and you can watch microtubules polymerize and depolymerize.
Microtubules continually grow from the centrosome added to a cell extract.
I had that on mute, and it's...
Quite suddenly, however,
all right, well, listen.
Stop growing.
Is that sorry, it's so loud.
Behavior called dynamic instability.
It will not let me control this at all here. Anyway, it's very cool because what you could see is at the same time some were growing and some were shrinking. So it's a really exciting question in polymer dynamics, you know, how you get the population to be simultaneously doing both of those processes. That was in vitro, and what we'll see here is watching it in a cell. This is going to look a little different than the movie I showed you here, because in this movie it was in vitro and you were watching all of the tubulin labeled. Here we're looking indirectly at the microtubules using a probe called EB1 that just binds the tips of these microtubules. So you won't see until the end of the movie the whole microtubule. You can infer that it's there if you look. You can see the trails behind it. But you'll be able to see the ends of the microtubules like fireworks here.
EB1 is a protein that binds to the GTP tubulin cap at the growing ends of microtubules. Cells expressing a GFP-EB1 fusion protein reveal the spectacular dynamics of the microtubule cytoskeleton. Note that many, but not all, microtubules in this cell grow from the centrosome. Only the ends of growing microtubules are visible in this experiment. Those that are static or shrinking have lots to their GTP tubulin caps and do not bind EB1. In contrast, when all microtubules are labeled with GFP tubulin, the true extent of the microtubule cytoskeleton emerges. Both growing and shrinking microtubules can be observed.
So in those movies, what you saw is this polymer assembling, nothing going on at the minus end, but at the plus end, plus for faster growth but also faster shrinkage, you're seeing outgrowth and shrinkage all the time. So again, cells are never static, and a single microtubule might be there, and then if you look 10 minutes later, not there, because they're continuously probing their environment, growing out, and then rapidly shrinking. So we think about these microtubules as tracks or as cytoskeleton, but it's really important to remember that they're also very dynamic and able to continuously remodel to fit their environment. So in an interphase cell, one that's not dividing, you see a beautiful net of microtubules as shown here. And then, of course, you all know that that microtubule cytoskeleton completely breaks down and then rebuilds for a dividing cell, as shown here. So this kind of dynamics is absolutely essential for a cell to divide, as well as for all the trafficking that we were talking about previously. And the importance of it is such that drugs that specifically block this process, such as Taxol, are used as anti-cancer drugs. This is one used for advanced breast cancer and ovarian cancer. Just stopping these dynamics is enough to stop cell division. All right, so for the rest of the time, I was going to go away from this sort of generic viewpoint about how things could work in a generic cell, and then talk about what real cells look like. So as time permits, we'll talk about 3 different examples of how these kind of dynamics can work in different cell types. We'll talk about neurons and think about issues of time and distance. We'll talk about muscle cells and think about a little bit about force and mechanics. And then epithelial cells, we'll think about their change over time. And again, keep the questions coming, because this is sort of a broad overview of cell biology. So first, what is a mammalian cell, or an animal cell, as Ram charged me with? In most labs, if you had one cell in common, it would be something called the HeLa cell. This is HeLa cells grown in culture. If you have time this summer, read this book. It's really fascinating. Have you guys heard about it? Anybody read it? Yeah. I think it's a great read. It talks about the derivation of these cells, and it also really is interesting in terms of insights into the early development of the biomedical enterprise, and also socioeconomic issues and legal issues. And it's just a really good read. But basically, these are the cell type that's favored by many labs because they're incredibly easy to grow. They grow like weeds. They're easy to use, even in the summer research project. They have all the elements that I've been telling you about. Here we're looking at microtubules, and you can see the nucleus in blue. Here you can see that common centrosome, sort of in the midst of all this spaghetti and the microtubules going out. If we added probes for the ER, the Golgi, they'd all be there, and we could watch all these dynamics happening. But these are a highly transformed cell type and don't really mimic any true mammalian cell type that you'd find in an organism, and that's something that you want to know. So to ask different questions, cell biologists in the lab will use many different types of cell lines or primary cells. Here's just a few different cells that have been used in my lab. This is from one of my students. HeLa cells stained for actin. This is beautiful because you're looking at really this plasma membrane-associated actin that we were talking about. COS cells, which are gorgeous. They're from African green monkeys. Like, who cares about that? But they grow beautifully large and flat, so they're great to do microscopy on. And then these are primary human cells, and so you can see in some ways they look like these cells, and they're some ways distinct. In fact, cell lines have proliferated so much that it's now a question about how you can differentiate among cell lines. So one of the guys in my lab, Jeff Nerschel, who'll be doing some of the one of the labs with you, has been interested in using machine learning approaches to be able to discriminate between different cell types and different organelles. So this is ongoing work in my lab where we're using machine learning to be able to feed images of cells in and then use unsupervised phenotypic profiling to discriminate between different cell types. And in these initial experiments, where we look at different cell types stained for the same markers, we can find that different cell types will group together in clusters. So with no information, the computer is able to recognize different cell types like the HeLa cell I was telling you about, or U2OS cell, and discriminate them to different clusters. that's probably not going to be the major importance of this study. What we're really going for is being able to discriminate other features of cells, such as those organelles that I told you about. And this is definitely ongoing, so as you can see, it's not perfect yet. But we're able to discriminate different organelles in the cell just based on machine learning approaches. So ask Jeff questions about that if you're interested in machine learning and deep learning. Back to real cells. Cells in your body rarely look exactly like those HeLa cells that I showed you in culture. And some of the most interesting and beautiful cells form these gorgeous and very specific morphologies. So the one I'm going to start with is the neuron, where you have that cell body with all these beautiful long projections that reach out. After that, we'll talk about muscle cells and how they develop in culture to have a very different type of morphology. Right away, you can already see this long cell with lots of different nuclei in it. And then at the end, we'll talk about skin cells and differentiation of skin cells. So first to neurons. Even if you didn't care about how the nervous system worked, even if you were purely a biophysicist or an engineer, you should be interested in a neuron, because you had this unbelievable problem. This is the dendrites, and this is the axon. And if you just look at this sort of schematic cell, there's nothing too exciting about that. Yeah, this axon is longer than the dendrite, but not incredibly much longer. But that's not how neurons are. Some neurons are like that in your body, but others are like this, which is a human motor neuron drawn to scale. This is the cell body. This is the tip of the axon. So from here in your spinal cord to your toe, a single cell that has to live for your entire life. This is where the proteins and new organelles are generally made, and they have to be shipped out all along this, all along this, all along this to get to there. So now you start to see why it's a really interesting engineering problem, and why this long-distance transport that we've been talking about a little bit with motors is so important. What kind of cargoes are being shipped out are pretty much all these organelles that we've been talking about. Endosomes, lysosomes, degradative organelles called autophagosomes, and mitochondria, which is the ones we're going to focus on. And the way they're shipped out is along the microtubule cytoskeleton in vesicles or organelles driven by molecular motors. So what does that look like? Oh, wait, first, just to talk about the timing involved. Anyone could say a meter is a long distance when you think about a cell. But if you want to put a time on it, that's where it becomes really amazing. If you think about the distance it takes to ship cargoes along something like this, well, even a millimeter would take a few minutes. But if you're talking about a motor neuron, you can right now be making new proteins in the cell body and your spinal cord, and that will be delivered to its site of need at the earliest in 2.5 days. That's what's called fast transport. It turns out your neurons also have something called slow transport, which means new proteins won't be delivered for months. Yeah, so and this is the business end of the neuron in terms of communication with the muscle. So this is not just like a distant part of the cell you don't care about. This is like the critical part that has to work. And it requires active movement, continuous movement of organelles and proteins to this axon terminal. So that's why people like me are obsessed by this question. So as I said, it's driven along microtubules, and we already discussed these polymers. The only difference to microtubules in the axon here is they tend to be a bit more stable than the ones that I showed you. I showed you the ones that are continuously dynamic. The ones in the axon tend to be bundled a little bit, sometimes bundled, sometimes not, but generally more stable, because you wouldn't want these tracks to be disappearing over time. The motors that are driving this are specifically in the kinesin family, or an even more complicated motor called dynein, cytoplasmic dynein, which interacts with a protein complex called dynactin. it's a beautiful story that the kinesins take the cargoes out and the dynein takes the cargoes back in. So it's a very organized, non-random process. What do they actually look like? This is a beautiful movie of kinesins stepping along a microtubule. Did anyone play this for you yet? I don't know. Ron probably showed you. I'll show you again just to compare to the dynein, which he probably didn't show you because that's not in plants. So kinesin is this beautiful organized enzyme where you have, sorry about the sound effects, finding of one head, then allows the second head to release and throw forward. And when you look at this, and stop laughing, and which will be a bit, what you see is this is exactly the kind of motor you would want to have take a cargo out for a meter in length, right? Because if you look, one foot is not releasing till the other foot is firmly on the microtubule, right? So it's stepping like this in that movie, stepping like this, stepping like this. That's how you want it to work. When you see dynein moving, you'll be very nervous about your neurons. This is right now our best understanding of how dynein walks along a microtubule. Scary and wobbly. Unbelievable it could even work. It's humongous and complicated. But those are the enzymes that are moving these organelles we talked about all along the axon. One organelle that we'll talk about in particular is mitochondria. Hopefully it's a little bright, but you can see these mitochondria along here. Mitochondria are what you learned in high school biology, the powerhouse of the cell, where they're generating ATP, and you need ATP all along this axon to keep your motors working and all the other aspects of the neuron working. If you look carefully at this movie, what you'll see at any given time, some are stationary and some are rapidly moving. And that all appears to be highly regulated. Yeah. Oh, that's very cool. I'll go back to the movie, but I won't play it. If you were watching this really carefully, and I can show you later, there's a specific change in what's the neck linker here. Once this head binds and you get a change in the nucleotide state here, that neck linker changes its orientation and becomes like sort of glued along here, and that's what swings the other head forward. So they're tightly coupled kinetically and actually somewhat structurally, like they're tightly coupled right here so that it will inch along. So that makes perfect sense. In terms of dynein, these heads are much more weakly coupled, and they have a much higher chance of getting it out of sync, so it's kind of like this. And you hope this isn't videotaped, but that's how it moves. And if you have the enzyme and look at a single molecule level, you can even show that it can move backwards occasionally like this. So what we now are starting to learn is that dynein itself is not a gorgeous, smooth motor like this. It will take wobbly steps, but it's constrained by different binding proteins to keep it behaving in a more processive way, meaning once it binds, it runs along the microtubule. Just to make it more complicated, these diagrams are accurate. At any one time, you'll have multiple different types of motors on an organelle. In this case, kinesins and dyneins, and they have to be regulated, one turned on and one turned off, but we don't have time to go into all that complication. So I showed you the mitochondria moving in neurons in vitro, meaning growing in a glass dish. You can also see that type of movement in vivo. This is from an explant. If you take a piece of a neuron, like a chunk of tissue out, you can watch mitochondria move as well. This movie plays. Nope, you could if it played. But here you can see what happens. You can watch them move. And people are now doing this in vivo. You can specifically watch them move, both in models such as worm or fly, but also in mouse. You can watch it live. Why does it matter? Because mitochondria have to both move to sites of need and then become stationed there so that they can locally deliver ATP when it's needed. So I talked about neurons as an example of why we can't just think of a generic cell. We have to think about cells very specifically if we want to go deep into their function. And with neurons, the major challenge is this distance and then the time it takes to get new material there and old material out. For muscle, I put this example in because, one, it's really cool to watch, and two, it makes you think about force and mechanics, which is a subject of interest to a lot of you. So many of you probably don't know this, but when your skeletal muscle is developing, it starts as myoblasts, single normal-looking cells, similar to like what we saw a helocell look like. But what happens as this muscle cell develops is these cells fuse to form a myotube, where you have a long tubular cell with many different nuclei in it. And then that myotube will then mature to become actually a myofiber that's activated by the motor neurons we were just talking about. So this can be modeled very well in culture. Here we're using a cell line, but you could also use primary myocytes and get this to work. And basically you can watch two different single myoblasts fuse to form this proto-myotube. And then as you go on in culture, these are the days in culture from day 3 to day 7, you can see more and more cells fuse together to give you these long tubes. And when you look at these long tubes, you can see in some cases the nuclei seem to be reasonably spaced out. In other cases, you see them somewhat clustered together. Well, a few years ago, I had this fantastic student in my lab who was obsessed with muscle, and she wondered what the movement of those nuclei looked like. And can you see this? We'll see if you can see it with the lighting. But basically, here we're looking at the nuclei in these myotubes, and instead of that classic picture where they just stay still and don't do anything, you can see they're like incredibly dynamic. You can see that some are just rotating, spinning like wheels. These are all clustered together, squeezing each other, changing morphology. One of these wants to pass, so he's squeezing by here. So we just thought that was unbelievably fascinating. And we wondered how that process could possibly work, because these are very large. So not like the little vesicles I was showing you, which are about, say, 90 nanometers. You know, these are 10 microns across. There's our scale bar. So at least 10 microns across, and yet they're incredibly active within this muscle cell. So a little hard to see in that phase movie, but here you can really see it very well when we start to put in different labels for the nuclear membrane, either inner or outer, or the nuclear lamina. And then you can see the dynamics really easily. So you can see them rotating, turning, spinning. We just thought that was amazing. You can track them over time and show that they rotate as an intact structure. And then we can look at the cytoskeleton and see if they're moving on microtubules or actin. So here we're looking at the microtubules in these myotubes, and we found that these microtubules are running along the long axis. And it turns out this movement is entirely dependent on microtubules, because if we put in a drug that causes the microtubules to depolymerize, a drug called nicotazole, instead of getting these beautiful long-range movements, we see no movement or hardly any movement. So then we thought, okay, if it's microtubule dependent, I wonder if it's dependent on that same kinesin motor that I was telling you about before. And guess what? Their kinesin is all over this nuclear membrane. So we used a cell biology approach called knockdown to deplete kinesin from these cells. And we could then look at the rotation and the translocation of these nuclei, and we see that if we inhibit the expression of kinesin, we see that they stop spinning and stop moving along the muscle cell. And what does that do? If you have no motor on there to drive their movement, they all wind up clustered in the center like this, which is similar to what's seen in some disease phenotypes. So the same motors that I told you about that have a role in moving small organelles along a neuron are also required to give you this spacing of nuclei along the long axis of a muscle cell. So very different scale, very different forces involved, because these are so large, and you could actually see them like compressing and moving as they're moving along. But the same motors in the same cytoskeletal system. So that tells you a couple of things. That tells you that the cells are using a similar mechanism, but in a very different context to do a very different cellular job in a specialized cell type. And now I'm over my time. I just want to tell you one other mini story, and that's, so far we've been talking about single cell dynamics, but it's important to remember, and Rick will remind you of this, that many cells, their major function is to function in context. And so increasingly, I think cell biology is going towards modeling these cellular interactions in systems such as this, where you can grow skin cells, primary human skin cells in a dish, and actually form skin layers that exactly mimic what you would see in vivo. So this is a postdoc in my lab, Corey Simpson, is able to do this, where you can watch these cells cultures growing in vivo and forming these stratified layers of skin, and then you can directly compare them to what you would see in human skin and model different processes in a three-dimensional way in which cells are changing over time. So hopefully I can give you just a little taste of cell biology. I was talking about flow and compartmentalization, dynamics with this change over time, and the ability to put large forces on things like a nucleus. And then the key idea that there's no generic cell in the body, that you get very specialized cells with very specific functions and morphologies that adapt common mechanisms to do different jobs. So that's all I have. Thank you for listening. If you have questions, let me know. Question down. Oh, there we go.
So this goes back to like the pathways you were talking about. You mentioned that they're like highly organized. Have we like discovered any factors that go into like what may drive their structure? And then also, has there been any studies that have looked at maybe the same cell and its structure and see if that's consistent in a static basis?
Yeah, so those are all great questions. In terms of the structure, the most exciting research there perhaps is going on in the ER that I was telling you about, and this idea that it's all tubes, and they're very specific proteins that form three-way joints to join those tubes, so we can know in distinct molecular detail how that structure is organized. But then it's changing over time, so we have to understand not just how it came to be, but then how it reassembles, disassembles, and reassembles. So I think that's very exciting work, and I didn't have time to go into the details there, but yes, we can know. Something like the Golgi is another case. It turns out if you depolymerize the microtubules, I told you the Golgi is in these discrete stacks near the nucleus. If you depolymerize the microtubules, the Golgi flies apart, and you'll see, if you looked at a Golgi marker, you'd see Golgi elements all around the cell. But weirdly, they still form these mini stacks where they still have a very vectoral process of modifying, processing proteins. So even though they're discrete, they still know, which is one face versus the other. They're still polarized. So yes, so for each case, we have to dig deep into the molecular mechanisms that allow us to understand the assembly, just like we did with actin filaments and microtubules. So great question. Yeah.
Is there a functional reason that the nuclei in the myotubes are so wild?
Yeah, so that's a great question, and I have to tell you, we went into this the backwards way. Instead of asking why, we just thought, oh, this is really cool. Let's look at what is going on and how come. The answer probably is because when the myotube matures, you need the nuclei to be reasonably well spaced along there. Why is that true? Possibly because these are very large cells, and each nucleus is going to put out RNAs and have local transcription and possibly be responsive to its environment, and you'll get translation and cloud. So possibly it's to evenly distribute new material being made. Possibly. Possibly is more interesting than that, and they're responding to signals in different ways. But we do know that when you have certain muscle diseases, you lose this distribution of the nuclei. They become more centrally localized. So it is a dynamic process, and we don't fully understand it, the whys.
Do you know if there's any, like along the same lines, do you know if there's any gravitropic effect of the nuclei rotating, spinning?
Oh, yeah. So I don't. We thought, oh, it'll be cool if it's always the same way, a Coriolis effect or something. But it turns out we looked at it. It seemed to be relatively random, and they'll switch directions also. So you'll see them spin one way and spin the other. As best we can tell, the spinning is a byproduct of their translation. But what we wanted to know, and maybe engineers could tell me a little bit better, it occurred to me that the spinning may fluidize the system. So if you're just taking a large, huge nuclei, and if you saw, they were almost like the diameter of the tubes, and you're trying to squeeze it along, you know, it'll take a lot of force to move it along. But if it's spinning and kind of, they seem to roll past each other. I don't know if you... What we really wanted to know is if you needed the spinning to sort of fluidize it so you could kind of roll past each other. And so what we were trying at the molecular level was to find a way to stop the rotation without affecting the translation. And we couldn't find a way. We tried a couple of different ways. We thought maybe the kinesin motor that I told you about is driving the translation, and I didn't tell you, but dynein is also on the surface, so we thought maybe dynein was driving this rotation, but we weren't able to answer that question. It still bothers me, but it's so cool, right, how these like squeeze by and everything. Yeah, well, there was a question over here first, and then I don't remember who had their hand up before.
I'll just go for it.
Yeah.
So, I know in other cells, like one of the things that happens is that you have these like Nesprin molecules that link your
Exactly.
to the actin cytoskeleton. So are those absent in myotubes?
Nope, this is a Nesprin-based function, yes. So these are specifically the motors hooked to the Nesprins. So Meredith, who did this, showed a direct link between the kinesin and the Nesprin and mapped the binding domain. So, yes, exactly.
It's very related to that question. So all of these Nesprin molecules are part of the link complex as well. All many cells also have expressed these molecules and these proteins. So I wonder, have you seen this phenomenon in other cell types, like perhaps cardiomyocytes that also fuse cells and have multiple nuclei?
That's a great question. So that's actually how we got into this. Meredith was obsessed with muscle cells, and we had done a previous study where we showed that in fibroblasts, as fibroblasts were migrating, the nuclei rotated. But back when we're looking at the fibroblasts, the rotation we saw was always in the plane. It was never this three-dimensional rotation that you can see in these muscle cells. So that's why we went into this muscle cells to look at rotation. We have not done it with cardiomyocytes. I haven't gotten anybody muscle-obsessed in my labs since Meredith, so we haven't gone back to look at that. But I think that's a really interesting question.
The question is, have you seen any functional defects or the clustering of nuclei in the myotubes, like what does influence phenotype?
Yes, so other people have done that, not us. There have been mouse models now affecting the Nesprins, and they do, you see this aberrant clustering. It's hard to, it doesn't have as striking a defect in the mouse. mouse as you might expect. So Yeah, so it's hard to say, okay, if they're not well clustered. What I was wanting to do was to see if we could see changes in gene expression. If you have big ideas, see gene expression and maybe like a biomechanical effect of the gene expression in the nucleus and if they're misspaced. But we never were able to, I don't know, that was all pie in the sky, so we didn't answer any of those questions. But good questions. There's a couple more hands. No, everybody's done. All right, thanks for listening to Cell Biology.